Hello, everyone. My name is Abigail Edwards, and I'm a student in the Bio 230W lab. And my project is on the effects of paclitaxel chemotherapy drug on BY2 cell mitosis. This experiment used two groups of BY2 cells to test if the induction of paclitaxel on BY2 cells will slow the cell cycle during the M phase. One group was the control, and they received no treatment, and the other group was the experimental group, and they were treated with paclitaxel. After completion of the treatment, both groups were fixed with Farmer's Fix and then compared to each other via analyzation. The results were analyzed and found to validate the original hypothesis. Cell division was reduced in the treated cells. Paclitaxel is also known as Taxol, and it is a mitotic inhibitor. A mitotic inhibitor reduces the ability of cancer cells and cells in general to metastasize by reducing the polymerization of microtubules. A mitotic inhibitor basically breaks down the mitotic spindle. And what happens is it stops the depolymerization. So therefore, the mitotic spindle cannot disconnect from the chromosomes. So thus you don't have the two separate sets of DNA that would go into the two daughter cells the way you would normally. Paclitaxel affects the cell cycle at the mitotic phase, specifically at anaphase, and Taxol binds to these microtubules to keep them intact and inhibit the replication. In a paper that I found by Wang, written in 2013, it mentioned that the cells were treated with paclitaxel undergo apoptosis, basically meaning cell death, via blocking the cell in the G2M phase. The use of paclitaxel on cells is important in chemotherapy and cancer treatments because it can stop the spread of the cancerous cells and therefore can stop the spread of the illness in an individual's body. So here's the cell cycle and the stages of mitosis. Specifically looking at anaphase, this is where Taxol has the greatest impact on the cell cycle. In anaphase, if you look at the numbers 3 and 6, you can see 3 is outlining where the chromosome, the DNA, is, and 6 is the microtubule that is attached to it. These stay attached to it and don't separate, thus inhibiting the cell from entering telophase. This is the main action of Taxol and of this drug. So it keeps the cell in anaphase for longer than the cell would have been normally without the presence of the drug. In this figure, you can see the growth percent during each phase of the cell cycle without the presence of Taxol. So normally you would have a lot of growth and a high percentage in G0 and G1. This is when the cells are normally growing, when they're normally replicating. This is in a normal functioning cell. In the second figure from the paper by Wang, written in 2013, the growth percentage during each phase of the cell cycle with Taxol, you can see has dramatically changed to the G2 and M phase. And this is due to that prolonged time spent in anaphase and cells not entering telophase. That's because the cells are just basically sitting there and they can't move forward. And so you have some cells that undergo apoptosis, and then you have some that just enter telophase a lot later. Either way, slowing and inhibiting replication. So the methods for my experiment is we have 5 milliliters of BY2 cells that are split into a control group and an experimental group. Both groups get 2.5 milliliters of these cells, and each group contains 864 cells specifically. The experimental group receives 5 milligrams per milliliter dosage of Taxol. This is something to note that is important is during normal human cancer treatments, humans are treated with 6 milligrams per milliliter dosage of Taxol. However, 5 milligrams per milliliter is what's sold via Thermo Fisher for $128 and nothing larger than that. So since we are a college biology lab, it would make sense to use the 5 milligrams per milliliter instead of having to order 2 and doubling the price. And for this experiment, the 5 milligrams will be sufficient to get the job done and to be able to see the results. Another thing to note is that Taxol is purchased in the liquid form. That is because this is already an intravenous medication. So when you are purchasing it, it's already in the liquid. So there's nothing more to be done there. Now both groups will be incubated at 28 degrees Celsius and 120 RPM. And then both groups will be fixed using Farmer Fix and observed using transmission light and fluorescent microscopy. So here we can see the results for the control group specifically. You can see that a lot of cells spent a lot of time in interphase, a good amount in prophase, and then it starts to dwindle down around metaphase, anaphase, and telophase. Specifically focusing on anaphase and telophase. You can see here there's some cells in anaphase and then some less in telophase, so it's already started to minimize. However, there are still more in anaphase than there are in telophase. And now, when we compare that to the experimental group, you can see that anaphase is now almost identical to the amount of cells in metaphase, and telophase has shrunk significantly. This is due to the prolonged results that anaphase has more cells in it because of the reduced mitotic spindle breakdown. So therefore, cells are spending more time in anaphase because they're no longer able to enter telophase as easily as they were before due to the presence of Taxol. So here are those results basically written out in numbers. So you can see that interphase, prophase, and metaphase all have the same percentages from the control group to the experimental group. And then you can see in bold where anaphase had 5.5% and telophase had 2.7% in the control group. Well then, when we put Taxol on the experimental group, you can see that anaphase went up to 7.63% and telophase went down to 0.69%, meaning that less cells were entering telophase than they were in the control group. The results support the original hypothesis that paclitaxel will slow the cell cycle during the M phase. The mechanism of disruption is the binding of Taxol to these microtubules, causing them to not be able to dissociate from the chromosomes. This then inhibits replication. The treatment slowed the cell cycle specifically during anaphase, and the mitotic spindle stays intact and doesn't shorten. Less cells enter telophase, and here is another summary of the numbers that we saw. The use of Taxol in cancer treatments is very common, and this is a good treatment that they've seen results with. Cancer is characterized by uncontrolled cell growth, so by using Taxol, doctors and researchers are able to get somewhat of a control on that cell growth and be able to reduce the unwanted cell growth. A problem that may arise with this technique is the potential to calculate the dosage of Taxol incorrectly. So as I had said before, the common treatment with Taxol is 6 milligrams. However, if someone was treated with only 3, or say 2, or they were treated with 10 or 12, more or less is going to cause problems, and either it's not going to be as effective and you are not going to see the reduced cell growth like you had wanted to, or it's going to be way too much and it's going to start targeting the cells that are good and targeting the cells that are helping that human to survive. So the dosage is very, very important for this, not just for our lab that we're doing today, but also for just treatment all across the board for patients and maintaining the safety and their healthy lives. Here are the references for the paper I used and the lab manual, as well as where I got the data. Thank you very much.